Hey there, welcome back to my channel. And today's topic is much more interesting. That is chromatography. It is a part of chemistry. So first let's have some question session. The question is, does black ink contain only black color or any other color as well? I mean like if you bring something black ink and you see that it's in black color. So, do you think that there's only black dye in that, or do you think that even there are many more dyes which are mixed in it? So, what do you think? Just comment me below. And the answer is it contains numerous dyes mixed in it, like blue dye, yellow dye, red dye, etc. These all are mixed together to form black ink. And how do we separate these as their properties are same and are present in very small quantities? Then how do we separate? That is what I'm going to show you in this video. We are going to deal with special separation technique that is chromatography. It is used to separate two or more dissolved solids which are present in a very small quantities. So if you see black ink, it contains numerous dyes in very small quantities. In order to separate those, we use this chromatography technique. And this chromatography technique, it is mainly used for the separation of mixtures, that is solid mixtures, which are present in very small quantities. And not only that, when the components even have very similar physical and chemical properties. And chromatography means writing in color, because chroma in the sense color and graph in the sense to write. And now let's enter into our activity, that is paper chromatography. There are different kinds of chromatographies for us: liquid chromatography, gas chromatography, liquid gas chromatography, and paper partition chromatography. For our grade, we are going to deal with paper partition chromatography. The aim of this activity is to separate different components of dyes using paper chromatography. And coming to the requirements needed: beaker, pencil, black ink bottle, filter paper or chromatography paper, and sellotape. And coming to the principle used: separation and identification of different components is based upon the different solubilities in the same solvent. So your solutes are the dyes and solvent is the water. Coming to procedure: first take a thin strip of filter paper, draw a line above the filter paper using the pencil as shown in the figure, and the outlet should be something like this. And put a small drop of black ink at the center of the line using the dropper. And let it dry for a minute. Now take a beaker and pour some more water in it. And now hang the paper strip with the help of a pencil and tape in such a way that a drop of ink on the paper is just above the water level. So as you see in this image, the drop of ink, it is just above the water level. Now place the paper strip for about 10 minutes. Observe the changes, and this is what exactly you're gonna get in your activity too. Now you're supposed to observe the changes. And the observations goes like this. The water gradually rises up the filter paper due to capillary action. And as the water moves up the step, it takes along the dyes present in the ink. And the dye which is more soluble in water dissolves first, rises faster, and produces colored spot on the paper at the higher position. What about the less soluble dyes? They dissolve in water a little later, rise lower, and produces colored spot at some height on the paper. So if you see in this activity, you can observe that black dye, it is more soluble in water for which it has reached something higher position. But coming to violet, it is less soluble in water because of which it has produced its colored spot at some height on the paper. Coming to the inference in this activity, we got to know that we can separate two or more substances present in the same solution, that too in very less quantity. Chromatography, it is a laboratory technique for the separation of mixtures, solid-solid mixtures into its individual components. And we can use chromatography to separate components of dyes present in the ink as we have done in this video. And the separation and identification of different components is based upon the different solubilities in the same solvent. As you have seen over here, the solutes are the dyes. The rate of solubility of the dyes in water is different. That is why we were able to separate it. The process is also used to separate the colored pigments in plants and flowers, and also to determine the chemical composition in many substances. Coming to applications, to separate colored substances present in dyes, as we have done in this activity, and natural pigments such as leaves and flowers. And to separate and identify the amino acids obtained by hydrolysis of proteins. It is done by liquid gas chromatography. To detect and identify drugs present in the blood of criminals in forensic science, when it is mainly used by the forensic yard. And at last, to separate small amounts of different products of a chemical compound, even when the physical and chemical properties are similar. And that's all up to today's topic. Meet you the next time. Before that, I'd like to say thank you for your patience.